Hey all, it's Dr. B. This is a part two of a video on protein structure. So now that you kind of have a fairly good idea about the generic folding aspects of protein structure, I want to talk a little bit more about something called a domain and kind of what that is. You look at this thing here, this is a protein called P53. It's a tumor suppressor protein, and its job is to basically either correct DNA damage errors or to direct the cell into apoptosis, which basically means blow itself up if it can't fix the issue. And there are some diseases associated with mutations. Liefeld syndrome is one. There's a bunch of others. But you look at this and you're like, this looks like a bowl of spaghetti. And from the last video, you probably can point out a few things, like maybe a few beta sheets and definitely some alpha helices and whatnot. But let's add some life to this, and that's to me what domains are. And so to kind of get a better sense of them, let's just kind of look at this protein and kind of what it needs to do, and then we'll talk about what a domain is and what its domains are. So this is the P53 tumor suppressor protein, and this is just a smattering of interactions. It's not all of them, but this thing directly connects with and activates some other protein, specifically this one right here called MDM2. You do not need to know any of this. I'm just giving you a sense of how most proteins really work by interacting with other proteins depending on what the cell needs to do. So now the question is, how in the world does something like this spaghetti looking thing actually do all of this? Well, it has parts of its body that do different things. And just like you and I have, fingers that have a certain way that they look and move and function, that's very similar to domains of proteins. So on that note, so let's take a look at the actual domains of the P53 tumor suppressor protein. Now, excuse me, first of all, what the heck is a domain? Generally, it can get complicated, but keeping it simple, it's basically a segment of a protein that can fold on its own and work independently from other parts of the protein. And the structural and evolutionary and functional units of proteins are another term for what a domain would be. And they're also homologous segments in evolution. So it's a great way for us to kind of be able to compare one way different proteins that might all have a domain that does a certain thing. So domains are basically units of structure and also in essence function of a protein. So remember all the crazy stuff that this thing had to do. So let's see if we can figure out a couple things from it. So first of all, let's start at the left. This is amino acid 1 to 42, and first of all, you can tell that this has 5 domains. P53 has 5 domains: transactivation, proline, DNA binding tetramerization, and a basic domain. And there are other areas here that also do some particular things. So now if we take a look at this, just from our last video, we see that there's the N domain, the core, and then the C domain. And so starting here, we have a transactivation domain. Well, that basically does exactly what you think it does, which is this would activate other proteins. And specifically, this makes sense that this would interact with, for example, MDM2 to activate another protein. And it also activates with other ones as well. And so that part of the protein serves that function. The proline-rich one, and again, you don't need to know these for like a test. It's just to describe to you how protein areas and domains do different functions. In this one, the protein-rich domain has to do with apoptosis. So if the cell, if it's attempted to recruit other proteins and to fix DNA damage and it can't, then this is basically the big red nuclear button, for an analogy. Obviously, we can tell something else about this, is that this is a DNA binding domain. So this protein literally binds to DNA, and that makes sense because its job is to correct errors in the S phase of mitosis. And so that would make sense that this would have to bind to DNA. And you can also imagine from some of the other presentations I've done that if there are certain mutations, missense mutations, where a different amino acid is put into polypeptide chain somewhere along here and it were to change the actual structure of this area, you can imagine it might decrease the DNA binding capability and therefore decrease the function of the protein as a whole. Let me finish up the rest of the domain. So tetramerization domain, that already kind of gives you a hint that this is a quaternary protein, and in fact it is. So as you learned in the last video, you have your primary, secondary, tertiary, and quaternary. P53 is a quaternary protein, which means there's 4 little subunits that are all glued together, and this is basically an area of the protein that has to do with gluing them together. The last one we have is a basic domain, and this domain, as you can see, is on the C-terminal end, and basically this is considered a regulatory domain, and kind of what that means is that this domain seems to be necessary for not only transactivation of other genes, but also function of a couple other parts of the protein as well. So those are the larger domains, and there's also smaller areas of function of this particular protein. This right here, by the way, is NLS. This is called nuclear localization signal, and this allows the protein to move into the nucleus, which makes sense. This one is the same, except it's an export. So this is a nuclear export signal, and this allows the protein to be exported into places where it might need to go, cytoplasm, etc. So all of the numbers have to do with the actual amino acids and which ones have to do with which domain. So now that you have a fundamental sense of what a domain is, we can now use the idea of domains to compare not only homology, but to find out, hey, which proteins happen to have DNA binding domains or tetramerization or whatever you want to do. And this is where a lot of the work in proteomics is done, because this is how we would need to figure out, well, how do we know that P53 interacts with MDM2? How did we do that? There's many ways that you can do that, but some of the ways would be just to kind of isolate that domain from the rest of the protein, and like a plasmid or whatnot, and do things such as like a yeast-2 hybrid or others type of EMSA assays and whatnot. But that's how we can kind of do this. So I wanted to just give you an overarching, secondary video to explain how alive proteins are, and not as boring sometimes as something that you see that looks like this. So thanks a lot.